Good afternoon, everyone. It's always an honor to welcome today's speaker, Dr. N.K. Singh sir, Project Director, National Institute of Plant Biotechnology, New Delhi. So I request my coordinator to please welcome both the dignitaries.
Thank you so much.
Now I request Dr. Sally Praveen ma'am, Course Director and Head Division of Biochemistry, IRI, to please introduce today's speaker.
Thank you, Dr. Ranjan. Welcome back after the tea break. Today we are having a very, very illustrious scientist from ICR, Dr. N.K. Singh. I am not going to the details of the CV given to me because he has so many awards to his credit. But I want to highlight certain things so that you can appreciate a scientist in his lifetime can do many wonder things if he is passionate about. So Dr. N.K. Singh did his PhD from Australia and then postdoctoral also from CSIR. In India, there are several science academies. I hope you are aware of science academies. So 3 important science academies are there, and very few scientists in the research career can get the fellowship of all the 3. And Dr. N.K. Singh is one of them. So you can appreciate his research passion. That is how he got the fellowship of all the 3 academies. The other important thing I want to highlight, in the previous lecture you heard that every research scientist want to publish his research in the journal Nature. So we have a scientist with us who has publication in Nature. So that is another important criteria, because I don't want to go to many awards. You can imagine yourself. And the third criteria I can say that in ICR system or in the research system, there is a called as chair or a national professorship, which is given only to eminent scientists who has contributed throughout their life in a particular area. And among national professorship, out of 10 national professor, one is called as B.P. Paul Chair, based on a very renowned scientist, Dr. B.P. Paul. So sir is having that B.P. Paul Chair also as a national professor. So based on these 3 things, you can imagine what type of scientist we are having with us today to deliver a lecture. And why he is here? He is a man in functional genomics. When we talk about, he has a lot of contribution in genomics also. But once you have genomics, it is good to have their function also. So how to relate genomics with the functional genomic is totally his research is dedicated in that area. Currently, he is a director of National Institute of Plant Biotechnology, which is in Pusa campus itself. And he is going to talk about wheat again. So you have several lectures on wheat, and this is just, you are going to enjoy his lecture. Welcome, sir, in this auditorium. And these 33 students from different universities, and this is a live webcast, so I feel many more students will get benefit of your talk. Welcome once again, sir.
Thank you so much for a nice introduction, and very good morning to all of you. The students coming from all over the country, from different universities and institutes. In the next hour, I am going to talk about a very particular topic, about a wheat protein which we identify, which we discovered. That means it was not known before. And then I'm just going to tell you that how we discovered it and what is the present situation with it. And I will also tell you a little bit about genomics. Before I start, I will start with the acknowledgements. The work I'm going to present, it was made possible because of the funding supports in the form of a postgraduate research fellowships. Like many of you will have fellowships I got from the University of Adelaide. Then after coming to India, I received funding from Department of Biotechnology and Indian Council of Agriculture Research mainly. A large number of scientists have been responsible, have helped or collaborated with me, including my PhD supervisor, Ken Shepherd, Peter Language from Adelaide University. Then from CSRO, Colin Wrigley, Greg Donovan, John Skerritt. Then from CFTRI, Dr. Rajgopal Rao, from Pantnagar, Dr. Garg, and from IRI, breeders Dr. GP Singh and Dr. PK Singh. They have supported this work. So it's a long journey. That's why I have given the title, The Story of Trypsin. And then also along the way, apart from myself, the number of students have also contributed. So you will see that how just to get a little bit of work done of significance, of course, it takes a lot of time and effort to do something. And that's why the persistence is very, very important and focus is very, very important. I also had the very excellent technical support in my Australian lab, which unfortunately we don't have much in India, and that's why we don't have much continuity. So this is the outline of my presentation. First I'm going to, because your course is on the omics tools and the improvement of nutritional quality. So I will just give you a little bit very brief introduction. What is genome, why we have to do the genomics, and an overview of what our institute and ICR has done in the area of genomics. Then brief introduction of the gluten proteins and how the trypsin were discovered and what is their role along with the major protein, which is the gluten protein, in bread and chapatti making quality, and what is the prospects. So I am not going to go into too much details of these things. It's a more of an overview and in a more summary form. The idea is to convey to you just that how these genomics tools can be used, what is the philosophy behind it, how we can use it, and then giving you one example of how from nowhere now we are in a situation where we can make use of this protein in wheat quality improvement. So if you have any question in the middle, you are free to ask. You can just raise your hand and ask, or even without raising hand also you can ask. So let us start with the genome. What is the definition of the genome? Now genome was defined almost 100 years back, now it is 2020. In 1920, a German botanist called Hans Winkler, he defined the genome as the complete set of genetic information present in a single haploid cell of an organism, including its protoplasm. That means it also includes the mitochondrial and chloroplast material. At that time, DNA was not known, but this is the overall encompassing definition of the genome, which is still true. So that includes the nuclear DNA as well as the protoplasmic DNA, which is mitochondria and chloroplast. Now, why do we have to spend so much time and effort to decode or to sequence the genome? That means you have to do the complete sequencing, from every base pair, and that is quite costly. At least it has been very costly. Now it is much cheaper than what it was earlier, but still it is quite costly and take times to prepare a genome of very high quality, which is called reference quality genome. The first use of genome is the very basic aspect, or the knowledge curiosity. Once you have the complete physical map, genome is a physical map. Okay? Genome sequence is a physical map, is the real entity which you can see with your eyes. Under microscope you can see that this is the base pair, and that's how the genes are made up. Whereas the genetics and the genetic map, they are not physical, they are abstract thing. They are just based on the recombination frequency, you tell that this is the distance between the two genes. But the genome sequence is actually the physical map. So once we have it complete physical map, then we can understand the evolutionary and phylogenetic relationship among different species. Now the lot of genome information is coming from microbes to the human. Many species are getting completely decoded, and when we compare these sequences, you can very clearly see that how these different species, they are related to each other, and how far back, how many million years ago they had the common ancestors. So it actually reaffirms the theory of organic evolution proposed by Darwin and so many other people, that these different species on the planet, they have evolved from common ancestors. After the evolution of life, then they have diverged, because there is no other way that you will have so much of similarity. So based on the how much the similarity and how much the difference is there, one can work out how far back they were common ancestors. Even the plants and animals together have the common ancestors. So that's our origin, that's our curiosity. We want to know that who we are and where we are going, and what is all around us, all the life forms, how it has come about. So that's very important. So science can give you the answer of your curiosity. But it's not the plain only the knowledge. And knowledge must have application, otherwise that knowledge has no use. If doesn't help you in everyday life. So the very first one is that you can use the genome information for high density fingerprinting of your germplasm resources. You understand what is the germplasm? For every species which we use in agriculture, or even in medical and other things, there are so many variation is there, so many in that population of that species, there are so many individuals are there, and they vary from each other. They have different kinds of genes which can be utilized for improvement or for so many other purposes, genetic engineering. So this germplasm, these are the actually wealth of a nation. Under the biodiversity treaty, and the countries have been given right over their germplasm. Because you will see that most of the developing countries are poor nations. They are rich in germplasm, but the developed countries, they are not so much rich, because it is indirectly proportional to the development. The countries who develop too much are within the country also. The states like Haryana and Punjab, which are very developed in agriculture, they hardly have any germplasm left. There is no wild or natural things are left. But if you go to Jharkhand or Chhattisgarh, you will find so many. So this is true at the national level also, and within the country also internationally. So the people who have preserved over the years their biodiversity, they have been given a right over it, because it is very valuable. It can be utilized for the well-being. It can be utilized for the business also. That's why you have to have a right over that. But how will you prove? If somebody takes your germplasm, unless you have very high density fingerprint, you cannot prove that that material belongs to you. But once you have it, you can prove. Then of course, in the process of gene discovery, what is the use of germplasm? What do you find in the germplasm which is useful? It is the genes. Nothing else is useful. Of course, you already have the information, knowledge base, but it is the genes which are going to be used, either within the same species or in other species by transgenic, and the same species by normal breeding. And there is a lot of variation. The genes, you find the genes, then you find the alternate forms of those genes, which could be quite variable also, that performs a particular function and affects a trait. So there having genome information is very, very useful to find the gene, because it is just like having a GPS. Nowadays everybody uses GPS, that I have to go to place A to B, and then you put in the GPS, it just guides you through. Without the GPS, you have to ask so many people, you have to wander around. People in early days, they used to get lost. And even lifetime, they will meet after so many years that that person has been found. But now with complete information like your Aadhaar fingerprint and GPS, one can really tell which person is where. So that is the advantage of having the complete map. The genomes are so big, to find a gene without this information is like looking for a needle in the haystack. And then finally, actually the breeding process itself, the selection process, which breeders use to select a variation, genome information is useful as a selection tool for the foreground as well as for the background selection. So these are the important points why we spend so much time and effort to decode the genomes, why the whole world is doing it, not only us. Now let's come to the more practical aspects. Whether it is a particular crop or any commodity, any animal or cattle or sheep, whatever commodity we are using in agriculture, what we are trying to get, we are trying to get more production, that is more quantity, as well as a better quality. You want the more of a better quality thing. That's why you have the improved breed, improved varieties with high productivity. This is our main goal is there, the yield and the quality. Now the yield and quality are affected by, it can be improved, or you can get higher yield and better quality by so many different ways. There are two important pillars are there. One is the plant breeding, that means you must have a better variety, and the other is the better crop management, which is called natural resource management. You have to give the irrigation, fertilizer, whether it is a biofertilizer or chemical fertilizer, doesn't matter, but it has to have nutrition, how you manage it, at what time you give the water. So a good seed with a good management will produce you more of a higher quality. Then also different kinds of stresses are there, biotic and abiotic, diseases and pests, and too much water, too little water, salt. These stresses actually affect realization of the full potential. You have to manage these stages either genetically or through management, so that you can achieve the full potential of the variety, and they can affect both yield and quality. So this is the overall scenario which we are working. Today we are going to talk more about the plant breeding, how you improve, how you develop a better variety or better breed of animal or better microbe for biofertilizers and biopesticides. This is the process where the genome information is very important. And it all starts from the biodiversity, because we have a huge biodiversity. We have so much, a huge population of every commodity, every rice. If you see, there are more than 1 lakh varieties are there. There are so many traditional varieties and wild rice. Similarly, in other crops also, in breeds also. So if you want to discover the gene must be discovered. If you want to do precise breeding, if you want to make the breeding more precise, then you should know about the genes, which gene is controlling a particular trait. Of course, you can also do the breeding without knowing anything about the gene. That's how the traditional farmers or hunters and gatherers became the farmers. They selected something which was better performing. Then conventional breeders also, they made the crosses, and from that they selected by trials, they found something better, and then without knowing about the genes, breeding was done. But nowadays, you know every base pair of a genome, you know how many genes are there, what they do, then you can do more precise breeding, you can do more precise selection in less time, and much better varieties can be released. So here, breeding with more precise knowledge is very important to speed up the breeding and to make the efficiency of breeding faster. So it all starts with the germ plasm, and the gene bank collections are there. India has the second largest collection, more than 4 lakhs entry in the world. So first thing we have to do is to discover the trait. These collections sitting in the gene bank has no value. You have to screen them, you have to evaluate. That is called high throughput tools of evaluation, something like Genomics, or you just develop a methods where you can evaluate thousands and hundreds of material and identify one material which possesses a particular trait which we need. It could be a better quality, it could be higher yield, it could be anything, all those traits which are important, which you want to get. Suppose if you want a variety with high protein, then you have to screen so many thousands of them and find one which has high protein. Similarly for disease resistance, you will screen and find a donor. That donor may have very poor yield, it may have so many other traits, that it may be poor, but it may be good for one thing. So what breeding does is that it combines, you take good things from so many different places and put into one variety. The good genes are not there in one variety, they are distributed across so many different varieties or genotypes. So the job of the breeder is, or geneticist or genomics or biochemist is to find those genes, and the breeder has to put them all together to make a super variety. So for that, first we identify the donor, then we go to gene discovery. So to find the genes, there are methods we have to use functional genomics tools. I cannot go into detail, there are number of tools are there which allow you to find the gene in a particular genotype. If the particular genotype has a particular trait value, one can find which is the gene responsible behind that trait. And that's a very important area of research. Many of us are actually not doing it. You have to go into depth. There are very few examples where the genes have been identified which govern a particular trait. We know that the gene is there in that material, we even mapped it, but we are very near it, but we are not quite there. We haven't found the actual gene. So those examples are very few. But tools and processes are there. Once you identify the gene, then every gene has so many different versions, alleles, what is called alleles. So alleles are what? They are alternate forms of the gene. That is the definition of the allele, allelomorphs. So one allele may be better than other, definitely, because if you remember the Mendel's, he had only two alleles for each genes, tall and dwarf, round and wrinkled, like that, yellow and green, pink and white. So that was very early days, we didn't know the DNA, he didn't even know about the chromosomes. He was a mathematician, basically. So he just worked on the theory A plus B square. So there were only two alleles, and then he got 1:2:1 ratio, which is A square plus 2AB plus B square. But now we know that at the molecular level, now you know that there are chromosomes, there are DNA sequences there is a variation. So present day scenario is that there is no dominant or recessive, there is nothing. At molecular level, everything is codominant. If it is there, it will express. If it is not there, it will not express. So recessiveness is usually loss of function. Then other thing is that every gene has not one or two, it could have many alleles. It could have hundreds of alleles. And those mutations may have a functional implication, they may not have a functional implication. So we have to find, actually we have to mine the alleles. For that you have to go back to the germplasm. You found one donor, then you have discovered the gene because of the contrasting nature of that gene and using functional genomics tool. Once you identified the gene, then you go back to the germplasm, see how many versions of that particular genes are there and which could be the better. Now this is an oversimplification because the genes, they don't work alone, just like a human being cannot work alone. Alone, single human being cannot do anything. You always have the group and the societies, then together they can do the thing. Similarly, genes are also like people. There are good genes, there are bad genes, they work together to perform a certain particular function. So we have to identify those interactions plus better alleles, then it can be used in the molecular breeding. Then only it is molecular breeding. If you are going with the knowledge that this is a particular gene, better allele, this is the marker for that, if I put it there, then this improvement will take place. Without that, it is conventional breeding. So now molecular breeding also has the so many tools. You can have a large biparental population, or you can have multiparental population. You can do genomic selection, marker-edited selection. So many other things are there. Then you go to the higher yield and quality, which goes into the society, which is our purpose. What is the purpose of your study? You are doing MSc, PhD in biotechnology, biochemistry, genetics, breeding. So what is the purpose? Why we are doing? Simple answer is that I am doing so that I can get a job. a नौकरी. आपको जॉब मिल जाएगी, आपकी शादी हो जाएगी, फिर बच्चे हो जाएंगे, फिर they will do the same thing. They will go around the circle. But the real purpose of doing this study is that you can be useful to the society. But you are not doing a charity to the society. If you are useful to the society, society is actually paying you for that, in lieu of that. Because you are useful, so you are being paid as a fellowship, as a salary or whatever. So how efficiently you are doing your job, but for the same pay, people are doing different level of contributions. So that comes your own satisfaction from that. If you do your job well, not only you earn your salary, you also get more satisfied that you have done your job well. You get appreciations from people. Not only you get your own satisfaction, but you perform better. So why society appreciates you? Because your work is useful for them. If it was not useful, then nobody will talk about you. So this is the flow of how the genomic information we will be using, and I'm just trying to tell you that it takes years go from here to there. There are many examples of that. It has taken 20 years, 25 years, 15 years. Even in the best of the labs in the world, it will take at least 10 years before a trait will go from the discovery to the utilization phase. So if one loses the focus, if one individual or one lab or one group, individual may have so many other priorities, but at least some groups, you be part of a group, or you make a group, make other people part of your group, which works on a problem for extended period of time, so that the output comes and it becomes useful to the society. Now, these couple of slides I spend more time because it is very important to understand the purpose of what we are doing, why we are having this training, why we are studying. The purpose is very important because that motivates you. If you understand why you are doing, if you are enjoying what you are doing, then you are happy and you are doing with more enthusiasm, despite so many difficulties. But if you don't know the purpose, if you are not enjoying it, then you will get under stress that why me? Why I have to work so hard? Why I am not getting this fellowship? How I will get job? All those kinds of things you will start thinking. So this is very important to understand, to go with a purpose, because you are a chosen one. These people whoever come here, you are studying in the best institutions and see so many lakhs and lakhs of people from where you have competed and come to this level. So it's better to go with a proper purpose and understanding to do something. And purpose is definitely not to, at least I don't believe that the purpose is to get a job and get a salary and raise a family. That is not the purpose. Because purpose is to be useful to the society. So let us come back to the genome, why it is so difficult to do the genome sequencing. Now we know that the genome sequencing work started in 1990s, the genome level sequencing, when the Human Genome Project and Arabidopsis Genome Project started. People were working on, they were refining the tools. It was very, very costly, more than $1 per base. Now it is less than 0.1 cents per base pair. But still it is quite cumbersome and costly. One of the biggest reasons particularly for the plant genomes is that the genomes are very large in size. They have huge size. There is variation mostly due to polyploidy and also due to amplification of the repetitive sequences. So whereas most of the microbial genomes will be around 5 MB in size, that is 5 million base pairs. If you go to the major crops like maize, barley, wheat, tomato, cotton, their genomes will be several billion base pairs, several giga base pairs. And wheat is actually 17 GB, 17,000 billion base pair, which is almost 6 times the size of the human genome. And you know that how difficult it has been to sequence the human genome. So it's mostly due to repetition in the genome. The number of genes are not that many, but the repetitive sequences are so many. And also there is a polyploidy in the plants. which makes it quite difficult to sequence the genomes. Because genomes are like jigsaw puzzles. You have to break them into pieces, and then after sequencing you have to join them together. The question is that why we break them into pieces in that case? Because the sequencing technique itself, it works on the pieces. You cannot simply go and start reading the sequence in the intact chromosome. You have to break them into pieces, then you have to sequence them, then you have to put them back together based on the overlapping sequences. So the very big picture, lots of pieces, very difficult to join. And a small piece with bigger pieces, easy to join. So that's why genomes like wheat and barley, which are very, very big, it will be very difficult to assemble them together back again. I will just give you very briefly just an overview of how India has been involved in the genome sequencing work. Before the rice genome project, India has not participated in any genome works, whereas internationally the genome work was going on, and all the major countries like Europeans, Americans, even China, Japan, they were partnered to that during development of the Human Genome Project and Arabidopsis genome, but we were not partnered. The first time we started in year 2000, when we joined the International Rice Genome Sequencing Project, and this project was supported by Department of Biotechnology and ICR again. We sequenced the chromosome 11 long arm of rice. So there are total 12 chromosomes. So by taking part into this international project, we learned a lot of things, and we became very proficient in doing the genome sequencing work, because we had to learn everything. So every country here, qualitatively, they had to be at the same level, because nobody was repeating other person's work. So there was a common standard was there. Everybody has to meet that standard. So it involved a lot of work, setting up the laboratory, learning the techniques, going for training, then coming back and doing here. Nobody did our work. We had to do our work, as a partner, as an equal partner in terms of quality, but depending on how many chromosomes one is doing, like Japan did almost 50% of the work. So this work, it took 5 years to finish. 2 years to generate the draft, another 3 years to make it actually reference quality. It was published in Nature 2005, and this is cited thousands of times. People make use of that information. So all the rice geneticists and breeders, and even in other people who are working in other crops, they make use of that genome sequence information, which is publicly available in the NCBI gene bank. So these are all public resources which were generated by the public through international collaboration, so that people can use it freely for their studies. Similarly, once we learned that thing, then we took the tomato genome work. This time we did the one full chromosome 5 of tomato. And this work was published in 2012. Now during the tomato genome, I must tell you, because now everybody talks about the NGS, which is next generation sequencing. So the rice genome work and the even human Arabidopsis, this genome sequencing work was done what is called clone by clone sequencing. You have to take the DNA, break into pieces and clone them in bacteria in using some kind of vector. Either it is a plasmid vector or bacterial artificial chromosomes, where the DNA has to be cloned for sequencing. And then each clone has to be picked up with using toothpick, then purified. So it was very cumbersome, and that's how it was costly. In about 2008, and these next generation sequencing techniques came. There are 3 different platforms. One is 454, and other was Solexa, Illumina, and similarly the other technique was there, solid, what is called solid, that was ligation based. So these 3 techniques, they did away with the cloning, because the cloning was not necessary. Now you can PCR amplify the DNA fragments. So the PCR actually replaced the cloning. The DNA fragments were immobilized on a solid support system, either on a bead or on a glass slides, and they were amplified there. That was the clone, and then they were sequenced there. As a result, you could sequence thousands of different fragments in parallel. So you can generate huge amount of data, which was very difficult to generate through clone by clone sequencing. So this reduced the cost and speeded up the work. So earlier in the beginning, we had planned to sequence only 20% of the tomato genome, the gene-rich region only, which is called euchromatin region. But once these new techniques came, then it was possible to sequence the entire genome, including the repetitive region as well as the gene-rich region. So both euchromatin and heterochromatin completely, more than 90% of the genome was sequenced with coverage. Then came the wheat genome. In fact, ever since the genome sequencing techniques were used to discover this human genome and Arabidopsis genome and rice genome, because wheat is a very important crop, of the probably the most important crops globally, and mostly it is more important for the rich countries than for the poor countries, because all of the Europe, North America, everybody grows wheat, and that is their main crop. It is called the stuff of life. It is called bread is the stuff of life. Just like rice is very important in Asia, wheat is very important in the Europe and Middle East and all those countries which have, where the Europeans have gone, like Australia, America, and other things. For them, wheat is most important crop. And now for us also it is very important, but it is second to rice only. So because genome was so big that 20 countries have joined hands together, and it has taken so many years to generate a draft. Now, it was possible to still share these chromosomes because we can sort these chromosomes out using flow sorting, laser-assisted flow sorting. So you can have chromosome-specific DNA. So that's how it was possible to share still, even with the NGS technology. Otherwise, with the NGS technology, you don't need to share. A single country can do it because you are not doing chromosome-wise, you are doing the whole genome shotgun sequencing. But in this case, it was possible to share because we have the cytogenetic stocks where you can have a separate, you can isolate chromosomes, based on their size. I'm not going into detail, but we were partner, we sequenced chromosome 2A of wheat. So we started, half of the chromosome, and in rice, then full chromosome in case of tomato. Then we took the complete full chromosome of wheat, but the size is much bigger. Size of the wheat chromosome is much bigger. The one chromosome arm is equal to the whole rice genome. So complete 2A chromosome size was almost as big as 2 rice genomes, and one tomato genome complete. So the draft was published in 2014, and then another after another 4 years. So it is so difficult, so big, as I told you, that because of the repetitive DNA, it is not so easy to assemble it back. And still now we have a reference genome, so-called reference genome of wheat, but still there are too many gaps are there. Still people are working on individual chromosomes to finish it to much better quality with less number of gaps. So there are many sequence gaps which are represented by unknown bases, which is called N. Apart from ACGT, you will have so many places, unknown bases. But nonetheless, it is very useful for the gene discovery and other work in case of wheat. Now I want to give you 3 examples of where these are through international collaborations. But there are many species, many crop species, many fruits, plants, medicinal plants, which are uniquely important for India, where nobody else will put their money, that we have to do ourselves. So we selected some species like that, like pigeon pea is one of them, which is grown entirely in India. More than 85% is grown, produced and consumed in India. No other countries does it, except for Myanmar and some African countries. They grow it just to export it to India because we had shortfall in production. But thankfully during the last year or so, last couple of years. Now India is producing enough pigeon pea by itself. So we need to know the genome so that we can do better breeding in pigeon pea, same as we are doing in rice. So we sequence the pigeon pea genome on our own without any international participation. Then using those genome information, we started mapping the genes for dwarfness and plant type, branching, maturity. You can see there are a couple of regions here in the genomic region on chromosome 4 and chromosome 5, which govern basically the so many traits related to the plant type. And now this new variety, the at least first dwarf variety of pigeon pea has been released from Pusa, making use of one of these genes. Among the genomic resources, because once you have the genome information, you can develop so many tools for doing the genome analysis. And one of these tools is that you can develop microarray chips where you can do high throughput genotyping. Like you can not one or two markers, you can do thousands of markers simultaneously, which is very useful for so many purposes, for association studies, for genetic mapping, and also for background selection, where you want to select the background, you want to see how much background you recovered. So there are a number of applications where you need their chips are very, very useful. So we have developed chips in case of pigeon pea, rice and mango. Here I am telling you that we developed a chip based on the single copy gene and agronomically important clone genes and disease resistance genes. So this chip has 9,600 genes and 62,000 SNP. So we call it 62K chip. And the good thing about this is that we have on an average 6 SNPs per gene. So normally SNP, you understand what is SNP? Single nucleotide polymorphism. At a particular locus, the nucleotide can be, there are 4 possibilities, A, C, G, T, but normally they are biallelic in nature. Even though theoretically there are four possible alleles are there, but practically only 2 alleles are there. There is a reason for that based on the probability. So the SNP genotyping, the disadvantage is that it is biallelic in nature, unlike the SSR marker where you can have number of alleles. But when we go for the haplotypes, so if you have multiple SNPs per gene, then you can have haplotype. That is the combination of those alleles. So that is possibility is 2 raised to the power 6 if there are 6 SNPs. So now the biallelic markers becomes multi-allelic because you are scoring a number of SNPs per gene. So you get the haplotype information. Haplotype is nothing but the allele of that gene, alternate form of the gene. So those forms may be associated with the trait. So these chips have been useful, and it is very, very highly successful chip, more than 99% success rate and call rate. You can see A allele, B allele, and these are the heterozygote. So for each SNP, if you have 96 individuals, you can see which one possesses one allele, which is the other allele, and how many individuals are heterozygote. So we create high density map, and we have mapped the QTLs for so many other traits also. I am not going into detail. Similarly, now we are using the NGS technology, next generation sequencing technology. So we had used 454 there in case of pigeon pea. Here in case of jute, which is another crop which is important only in India and Bangladesh, but very important fiber crop. We have sequenced the genomes and published it, and then we have identified a major QTL for fiber quality. Then also the mango. This mango genome is very interesting because it has a lot of heterozygosity. Now if you see that all the genomes which have been published in the literature, starting from the human genome to Arabidopsis to so many genomes which are published in the NCBI database, all those genomes barring the microbes which have a single DNA, circular, these are the diploid organisms. These organisms have a pair of chromosomes. So they like human have 23 pairs of chromosomes. Rice has 12 pairs. But all along the scientists have assumed that both the chromosomes of a pair are identical, or at least they have taken that kind of situation. Either you take a double haploid, in the double haploid both the chromosomes will be identical, or you take highly inbred line, because through selfing, you self in many generations, then also it becomes just very close to double haploid, because the heterozygosity decreases every generation, and then you have the homozygous line. So most of these sequences are based on, they took a line which are homozygous and they assume that they have only one chromosome. Means they don't assume that they have only one chromosome, that they assume that both the chromosomes which are coming from maternal and paternal, they are identical. But in the heterozygote crops like a mango or so many other tree species, maybe some insects and other thing, heterozygosity is a very common thing in nature. In fact, it has several advantages also. And in crops like mango, we have no double haploid, there is no tissue culture protocol. So we are bound to sequence a material which is highly heterozygote. So there are a lot of challenges to complete this sequencing, because mango has almost 2.5% heterozygosity. That means every 40 base pair there is a polymorphism, there is a SNP or indel. So that makes it very difficult to assemble it, because when you, the assembly is based on the 100% overlap, 100% match in the overlapping region. So if you have got one SNP or one or two SNP in between, that will not collapse into one contig. So the maternal and paternal chromosome, they assemble separately. So this work has been going on for quite some time, and Australian and Americans, they are also trying to sequence. But in the beginning, we found that the assembly was larger than the genome size, and that was the main reason, the high heterozygosity. But then this problem was solved. There are a couple of methods by which this problem is solved. One is that which we tried ourselves. We took the very long reads. So now let us introduce to the third generation sequencing. So we had the Sanger sequencing, clone by clone sequencing, which used to clone the DNA and sequence. Then came the NGS, where the cloning was replaced by PCR. So you still needed to amplify the DNA fragments. But most of these reads were very, very short. The NGS technology, they give very short reads of 100 base pair, 150 base pair. Only one of them, 454, it gives a slightly longer reads of, initially it was 100 base pair, but later on it became 500, 600 base pair, like that. But now we have third generation sequencing, which is called single molecule sequencing. So in this technology, you don't even have to PCR amplify. You simply isolate the DNA, high molecular weight DNA, and you can sequence directly. So here you can take 10 kilobase of fragments or even larger, and they take one DNA fragment, one DNA polymerase, and a mix of nucleotides, and then you can sequence in a single very, very nano-sized wells. And one of these techniques is called Pacific Biosciences or PAC biosequencing, which is the real-time sequencing. Basic principle is same, that you detect the base as they are added to the growing nucleotides. That PAC bio is very common nowadays. It has been refined with less errors. Another one is called Oxford nanopore, where you pass the single-stranded DNA through a pore, and then you detect changes in the voltage. So different base pair, 5 base pair combination, they give different signals. So that way one can determine the sequence. You don't have the polymerase there. You simply have a pore made of protein, and then there is a helicase is there which unbinds the DNA, and then it goes through the pore, and then through the signals one can. So these are the couple of technology. Even this Oxford nanopore, it still has so much error. It is getting refined. But the PAC bio, the error rate has been reduced quite a lot. So the good thing is that now we have techniques, and there are some more things are coming up where you can sequence the DNA without cloning. You can simply isolate the DNA and take it and get the sequence information. And you can also get longer read length, which is very important for sequence assembly. Longer read length means having a larger pieces of jigsaw. You may have a very big picture, but if the pieces are big, then it is easy to assemble. So that long read length is equivalent to that, you can say, or analogous to that. So what we did, we allowed a lot of mismatches. Like we go for, instead of only 50 base pair overlap, we go for 1000 base pair overlap. Because if your read length is 5 kb, 10 kb, then we can make overlap bigger. We can afford to have a 500 base pair, 1 kb overlap. And then you allow mismatches. We allow 10%, 15% mismatches, so that the chromosomes coming from the maternal chromosome and paternal chromosome, they will assemble together despite having this polymorphism or heterozygosity. So as a result, we were able to assemble this very highly heterozygous mango genome into 403 million base pair. The total genome size is also almost, so more than 95-96% coverage we have assembled the mango genome. But now there is another technique which is called, now people have started using a new technique which is called trio-binning. With the trio-binning techniques, you can separate the maternal and paternal reads. Like you isolate the genomic DNA, you do the sequencing, then you can separate the reads coming from the maternal chromosome or from the paternal chromosome. Because every pair of chromosomes, one is maternal, one is paternal. And if those two are sufficiently different, then you will not be able to assemble that. So during last 2-3 years, there is a technique called trio-binning. That means what is important here, that you need to have the mother and the father and the progeny. We need to sequence DNA of three individuals, both the parents and the progeny. So with the help of this sequence of both the parents, you can work out that in the progeny which DNA is coming from the mother and which is coming from the father. So we were actually quite lucky in case of Amrapali, because both the male and female parents are there in the Pusa orchard. But most of the other mango varieties, you don't know which is the parent, which is the mother, which is the father of the parents. So many other varieties, most of the mango varieties, it will be highly heterozygous. So that's why it was quite difficult for people who took any mango variety without knowing the two immediate parents of those things. So we have done that way also. Using trio-binning techniques also, we have separated the maternal and paternal, which is the Neelam and Dasari. Amrapali is a cross between a North Indian variety, Dasari, and South Indian variety, Neelam. So as a result, it is a regular bearer and so many other advantages, small plant size. And since both the parents and the progeny are there in the Pusa orchard, we were able to separate them. Another thing, we need the very high density linkage map. One of the Chinese Academy of Tropical Sciences in Zhejiang, China, they have developed a very high density map of mango. So we use this genetic map to anchor the genome of mango. And like this, see, this is the mango has 20 pair of chromosomes and total genome size is 400 MB. So each chromosome is approximately 20 MB. And you can see here 0 to 20 MB, this is the centimorgan, from 0 to 160 centimorgan. So these markers actually mapped on the assembly. So there is 1:1 very linear correlation here, you can see at the chromosome level. In the region of the centromere, the recombination frequency is low. So 10 to 15, for 5 MB, the recombination is very low, but for the other regions, the recombination is high. So similarly, all the 20 chromosomes have been assembled and the number of genes have been predicted. I'm just going fast because we have generated a chip for mango also, which has got 82,000 SNPs. And this chip has been used to do association mapping in mango, because mango has a lot of variability, very small to very large, different color, different taste. That's why it is called king of fruits. And horticulturists have done a lot of analysis of their traits at morphological level, but now with the genomic tools we are able to identify the genomic regions at least with very high large score which are associated with the, for example here, fruit acidity, whether the fruit will be very acidic, very sour, or it will be sweet. Then the fruit length, there is one more QTL has been identified. Similarly, the pulp percentage, the ratio of pulp to stone, these are very important horticultural traits. So if we identify these genes, then we can develop better mango varieties. So this was the first part of my presentation, giving you a general overview of the genomics and what our institute has done in ICR. In fact, our institute in this IRI Pusa campus is the main center for genome studies in India, including all the institute, whether it is agriculture, general science or medical or IIT. This is one of the main centers where the number of genomes have been sequenced. The other being the Delhi University South Campus, and now more institutes are coming up like NIPGR and MBRI, CMAP. CMAP is not there, but IHBT plus CCMB, they are also started doing, but not as much as we have done. So we have taken a lot of time actually. I will try to finish in the next 20 minutes. See, I am just going to tell you about the story of trypsin here. Now, wheat gluten proteins, you know the wheat is a very important crop of the world and it is used for making bread internationally. In India it is used to make chapatti or puri, all those kind of things. Maida is there, you can make different preparations. But the quality of wheat flour or wheat variety is very, very important. The genetic quality, the quality of those proteins which determine the actually wheat quality. Because different varieties, all the varieties may not be suitable for making the bread or chapati. Some are suitable for making biscuit, they may have very weak gluten content. The medium is required for making chapati and strong gluten is required for making bread. So wheat gluten is one of the oldest protein actually. I think in 1840 or something, this work was done on the wheat gluten. Gluten was isolated. This is one of the oldest protein which has been isolated anywhere. And this has this elastic property, you can see. This is the gluten here. So what happens if you knead the dough, like in your home, when you make the chapati or you knead the dough, if you wash it with water, all the starch will be washed away. And then what will be left is that the gluten, which will be very elastic. You can try at your home and you can pull it. And because of that, actually many people have the false propaganda in the WhatsApp groups that wheat has got plastic in it. Because every wheat has the gluten protein which behaves like elastic rubber. So when you wash up the starch, you will see that it has elastic property. Because of that property, the bread loaf volume, it rises and it's quite spongy. Even the bread, your chapati also puffs up because of this elastic property. And you can just feel it that when you stretch the chapati, it comes back just like a rubber. Because this night when you eat your chapati, you can see. If you stretch it, it will stretch and then it will come back, spring back. It is because of the gluten protein. They have the spring-like structure. They have repetition. So now after a lot of search, now it is known that the gluten proteins actually play the important role in making the chapati. So even in gluten protein, they were first analyzed in 1907 by Osborne. I will give you his classification. So it has two main components, gliadin and glutenin. So glutenin provides the strength and gliadin provides the viscosity, and the right balance of the two gives you the gluten. So if it is too strong, then also chapati will not be good. If it is too weak, then also there will be problem. So there has to be right balance. So the T.B. Osborne was a scientist, American scientist. which classified the plant proteins into albumin, globulin, prolamine and glutenin. This is water soluble, salt soluble, alcohol soluble and acidic soluble. In fact, this was the classification it was given, in the plants first, and then it went into the other things. So that the albumins are the water soluble proteins, globulins are the salt soluble proteins. Now I will tell you that because then when I started my work in 1981, we did not know so much that how, we knew that as a gluten as a whole is important for the quality, but this gluten is very, very complex. It has got gliadin and glutenin, then within the glutenin there are so many of them are there, so many gliadins are there are hundreds of proteins are there. So what is their genetics, what is their biochemistry, their composition, how they influence the quality, these were the subject of the research at that time. There still are to some extent, but a lot is known now. At that time you can see the SDS page which we are using quite a lot nowadays. It was just, this technique just came. Which year it came? By Lamley. You must be quoting in your papers. I think it was in 1975, somewhere that this SDS page electrophoresis came. Before that, you had starch gel electrophoresis. Nowadays you have acrylamide gel electrophoresis. Of course, SDS page was done on acrylamide gel. You can see this paper in Nature in 1958. I was actually born in that year. So science is progressing like this. So in 1958 there were new methods. Starch means you take the starch and you gelatinize it. You heat it up. It will make a, like a jelly. Then you pour it. Then it settles. Just like you make the jelly that is made of the starch. And then you load the protein in there and you separate and stain. So these kind of crude methods were available. So when I started that time, SDS page was just getting refined, and a lot of these gliadin proteins were being separated on the starch gels, which was very cumbersome. You have to take the starch and boil them. You have to wear a gloves of asbestos so that your hand will not, burn. And then you degas it because you get the air while boiling. Then you pour it. You have to circulate water around it, so because it will overheat and starch will melt. So it was so much difficult to separate the proteins at that time using the starch gel. When SDS gel came, then it became slightly faster. So I had taken a project to do the genetic mapping of these gliadins and glutenin, because the gliadin and glutenin were known at that time. They were not mapped. So I was trying to develop a method which we can avoid the use of starch gel, because SDS gel was just available. So can we do this separate the gliadins on SDS gel? Because at that time glutenin was separated on the starch acrylamide gel and gliadins were separated on the starch gel. So we have to do the mapping of both. Now what we knew is that the basic property of the gliadin and glutenin, because gliadins are monomeric in nature. They don't aggregate by disulfide bonds, and glutenins are polymeric because they make polymers through disulfide bonds. So you have to reduce the disulfide bonds, then you can separate the gel. So this was the situation here at that time. You can separate the high molecular weight glutenin here because their molecular weight was quite high and they are separate from the rest of the proteins. Now this particular bunch here is a mixture of low molecular weight glutenin, gliadins and also albumins, globulins, and you cannot see the individual bands so clearly. So in the beginning in 1980s and 81, only this part of the protein were getting studied, only the high molecular weight glutenin. So first paper started coming that there is a variation between the varieties. And there is one paper came which showed correlation with one of these variation with the bread making quality. There is a work from UK by Peter Payne that showed that. Most of the studies were focused on this. There were no tools to separate this one. Of course, you had the starch gel which was very cumbersome. So what I tried, I just tried one thing is that because they are monomeric in nature, so can I use the SDS page gel to separate the gliadins, I simply excluded the mercaptoethanol, which is used to reduce the proteins to break the disulfide bond. Either use mercaptoethanol or dithiothiotol you will be using to break the disulfide bond. So if I don't break the bond, then the glutenin will be still, you can see the background is streaking here because they are polymers of different size. They don't separate into individual bands. I have many pictures to very clearly explain that. Because of the shortage of time, I am just not showing all those things. But then we are able to see these gliadin bands here quite clearly, are much better than here. So this could be used for mapping the gliadins, because for mapping you need to have a mapping population, then you have to analyze 100 to 200 samples, which is not possible through starch gel. So while doing this, I noticed for the first time that there were this couple of bands were here. You can see there are 2 bands there, very faint you can see. Actually there are 3 bands, there is one below that which is very faint. They are roughly in the ratio of 4:1. So this is the first time we saw the trypsin, or anybody in the world saw the trypsin, because the first time anybody run the gel, SDS gel without mercaptoethanol, because normally it is taken for granted that it is part of the procedure that in the sample extraction you have to have SDS and mercaptoethanol and urea, all those things people were using to break the different bonds so that you will be able to separate the individual polypeptides. So for need of separating, finding an easy way to separate gliadins, I excluded them, then I saw these new bands here. Then we started investigating that what are these things. So once again, this paper was published in 1985 as a new class of these proteins. Then I used that method to map the, I will come to that thing, to map the 2 genetic mapping, like one parent has a different version, different alleles of that trypsin. There is a narrow and the wide bands, like a narrow gauge and wide gauge railway you have. So this in the population they segregated, and then we were able to map, because high molecular weight gluten was segregating, this was segregating. Then you can see that which part of the chromosome, how many centimorgans, they are away from each other using the recombination frequency. So I devised a method actually, which I will show you, where we can also separate the low molecular weight glutenin also. Because what I did, I cut only this portion here and then cut a strip and then load on another gel like 2-dimensional. Then you can separate both high molecular weight and the low molecular weight without interference with these gliadins. So as a result, I was able to map trypsin also and the glutenin, low molecular weight as well as high molecular. Otherwise, the normal SDS page which was being used at that time, this portion, all of things were overlapping. You could work only on this part. Now, in fact, before I go to the trypsin, which was a new thing, this was a new protein, this led, this kind of thing, just by you plan something, you plan an experiment, and then you might find something new. Because you can never plan for a new thing. You can do regular things and new things will come. Because if you can plan it, then it is not a new thing. So because we found this thing here, new bands, and we started investigating their nature, then out of this thing developed, the low molecular weight gluten also we can separate. And for this I can simply just take this part and reduce it, mercaptoethanol, and then run it again, then you will get these 2 things separated. Now, further in just 2-3 years, I developed this method where you don't have to do the 2-dimensional electrophoresis. Based on just the solubility itself, you can just extract out the albumin, then globulin. You take a single grains of wheat in different tubes, in Eppendorf tubes. You crush them fine, then remove or wash out all the water soluble, then salt soluble, and then alcohol soluble also. And then whatever residue is left, you can reduce with mercaptoethanol, and then you can get very good picture of the both high molecular weight and low molecular weight. So this proved a very useful tool to do a lot of extensive work on the low molecular weight glutenin, which were earlier inaccessible. So these are glutenin as important or perhaps more important than the high molecular weight, but at that time they were not analyzed, simply because they were overlapping and there was no technique to separate them and analyze large number of samples. So this particular publication, say we talk about the value of publication. This was in Journal of Cereal Science, which is not a very high impact journal, not like Nature. Still it doesn't have very high impact, probably 2 point something it is there. But you see here this paper has been cited 576 times in today's. It is competing with the Nature and Plant Cell. And it was done in 1991, but still every year at least 20 citations will be there for this paper. So if the work is valuable, it's a methodology or something, or new things, then, and this is not about the tritsin, this is a methodology which you can, this is only 3-4 page paper is only 4 pages. Because this allowed people to separate this LMW glutenins and such an important crop, so it received so much citations value. Now let us come back to the tritsin. So here in the case of tritsin, we knew that tritsin is again is not a monomeric, it is a polymeric protein. Because what we did, we just cut this whole gel and then reduced it in a with mercaptoethanol and then turned it like this, 90 degrees, like 2-dimensional, and did the electrophoresis. Then we found that where these subunits were going. And we could analyze their genetics also, everything. I am not showing that slide because these genes were located on the short arm of chromosome 1A and chromosome 1D. And this mapping also confirmed that they were located on the short arm, whereas they were away from the gliadins. This gliadin locus and low molecular weight, they are located in one place, and tritsin, this is located near the centromere in another place. Now, then I proceeded to do more analysis of those tritsin. The very first question was that whether they are storage proteins or they are soluble proteins like metabolic protein or other kinds of protein, membrane proteins. So biochemical analysis showed that they are behaving more like the albumin globulin because the gliadins and gluten, they have lot of glutamine and proline. That's why they are called prolamine, because they are rich in proline and glutamine. Whereas the tritsin, it was not rich in these amino acids. It was more like the rice gluten or oat globulin and other pea globulins. So this is more like the legume protein. It was a legume protein equivalent in case of wheat. It has become a minor protein because this prolamine has become major protein because of the gene copy amplification. So now wheat has more prolamine, but it also has some of these globulins. So this tritsin actually belonged to that category. This paper was published, then eventually a sequence was also published. Now to prove that whether it is a storage protein, you do the sucrose density gradient centrifugation, then isolated the protein bodies, and we found that this was present in the protein bodies. These are the electrophoresis of different fractions. So this was present in the protein body fraction. So that shows that it is a storage protein. Another property of storage proteins are that they are deposited in the protein bodies, in the storage bodies. So by that time, the protein bodies of wheat were actually known. Using electron microscopy, people have visualized the protein bodies in the wheat endosperm. You can see these are the wheat protein bodies. This is the starch granules. And within those protein bodies, there were inclusions, there were dark inclusion bodies. Okay? So for a long time, people did not know that what is in that inclusion body, whether it is a protein or something else. If it is a protein, then why you have the dark and light? So when we purified that tritsin protein and developed antibodies, which we eventually used to clone that gene also in the expression library, then you can see here that the inclusion bodies, they had actually trypsin. The trypsin antibodies, they hybridized with this thing. They also cross-hybridized with the glutenin also. There was some glutenin here also. So for the first time, we were able to see that this inclusion body in the endosperm, they were actually trypsin. So they were present in the same protein bodies where the major protein gliadin and glutenin is present, but as a dark inclusion body, this is a globulin type protein, it is present in that. Then of course I purified the individual subunits of that. There was a large subunit, there is a small subunit, they are joined together and then they form the dimer, and then this dimer forms the tetramer, which we see on the gel when we don't reduce it. Then sequencing was done here. Now this sequencing was done with using the old method of Sanger sequencing and radioactively labeled nucleotides. It has taken me 6 months to sequence that 1.5 kilobase. Nowadays, even in 1 week, you can sequence gigabase space using the next generation sequencing technology. So after coming back here, because this was a new protein and nobody had worked it, it has a lot of significance. This can be used to improve the nutritional quality of wheat. If you can increase the proportion of this protein in wheat, then it will have more balance of the essential amino acids. So we developed a project so that we can clone it and do genetic transformation to increase the copy number of in wheat. So DBT supported this thing. So there we are cloning and then standardizing the transformation regeneration protocol in wheat, because that time the wheat was not transformed. Then at the same time we also developed the near isogenic lines with different seed proteins to see that what is the relative effect of different proteins, because you have hundreds of proteins and how these allelic differences affect the quality of the now you have the tool to you can analyze these proteins, both the glutenin, gliadin and trypsin, all different classes of the proteins. So that was one of the aim in this project, that to produce near isogenic lines. Means we take one wheat variety called SD2329, which was popular at that time, now it is not so popular. So it has medium quality. So you, by back crossing, you transfer each allele. So you have glutenin, the number of 6 loci are there, gliadin there are 6 loci, trypsin there are 2 loci. So you have almost 1 dozen of the protein coding genes are there, and they have different alleles in different varieties, which you can see on the SDS page gel. So by crossing, back crossing and analyzing on the gel, you can develop lines which are differ for one band at a time. The genetic background is same because you have made back crossing and develop the near isogenic line, so that you can study the effect of these proteins in a common genetic background. Earlier you had a segregating population, so many things are segregating, so it was difficult. So this took actually a lot of time, from Pantnagar until here, several years. So we were able to clone this trypsin from different varieties and develop this first transgenic of wheat in India also. Also we improvised the method of transformation with biolistic, and we filed patent also. Now this I must tell you very briefly that we talk about the transformation, genetic transformation. We have two methods. One is the direct transfer, the other one is agrobacterium mediated transfer. So one of the disadvantages of the direct transfer using biolistic is that you can get multi-copy integration, and the success rate is also not very high. But with the agrobacterium you get this good success rate because it's a natural process of transfer. But it is genotype dependent. Some genotypes respond better, others they don't respond better. So what we did actually, we purified the BRE2 protein. The whole agrobacterium machinery, there are so many genes are BRE A, B2, G. There are at least 6 or 7 genes are there, which make sure that the DNA is excised, made into single copy. Then it is coated by the protein, and the pore formation takes place. Then it goes to the pore and enters the nucleus of the host. Now, when it reaches the nucleus of the host, when it enters the host cell, it is the single stranded DNA coated with BRE2 protein. Of course, there is one BRD2 is also there, but only 1 or 2 copy at the end of the protein. So what we did, we can do it in the test tube also. You have cloned BRE2 protein, you can produce the BRE2 protein, and you can make the DNA single stranded very easily by high temperature or by alkali. So we coated it and then we did transformation, and we get at least 6 times more transformation efficiency. So this could be used, but people don't use it actually. But it is published and nobody, because after that I left the transformation work. But this is something one can follow if you really want to use this ballistic and combine this with the purified protein. Then this important thing is that we developed these 42 near isogenic lines which differed for single protein at a time in the SD239 background, and we multiplied the seeds even coming after here in the pusa, so that we can see the effect of individual alleles of high molecular weight, low molecular weight, as well as the gliadin and trypsin. And this took quite a lot of time, lot of crossing, then multiplying, then again maintaining the lines itself is not so easy, because it seems this work is not very glamorous, because you have to cross, you have to go in the glass house, you have to make sure that plants they don't dry or squirrel don't eat it. At the same time, you have to check on the gel also that they are correct one. So these are the 42 lines to start with, and when I came to IRI, this was reduced to 31 only because of the maintenance problems, and there were no projects were there. But we have 6 nils per HMW proteins, 7 per LMW, 12 nils per gliadins, 4 per albumin, and these 2 per trypsin, because we had 2 variation. Now I have only 2-3 slides left so I will leave you soon. So you can see here we have actually, these are the nils, 31 nils, and 2 of these nils belong to trypsin. Like they are coming from the wheat variety K68, and the amount of protein is higher there. You can see on the gel that it produces more, not too much more, but more than other varieties. So we transferred, tried D1 and tried A1 separately. And then we grew in the replicated trials in Pusa field here only. You can see that there are 6 rows of each line, and you have 2 kinds of gloom. One is the dark gloom, other one is the light one. So this is because one of the gliadin genes on chromosome 1b, it is very tightly linked with the gloom color gene. So these actually this variety is the original variety here, SD2329. But when you put another gliadin locus, which is got tightly linked with the gloom color also becomes white. But otherwise, this very similar to, they are identical to each other. Then this work we published just 3 years back. Now it is 4 years. And you can see here all the green ones. I don't expect you to read it, that this trypsin here, trypsin D1 and A1, they had very strong influence on the bread making quality, both chapati making quality and bread loaf volume and chapati score. So just by adding this single allele coming from K68, now this is the allelic difference, like SD2329 also has 1 allele, and when we substitute it with another allele of trypsin, then the quality score becomes very, very high. Similarly, HMW glutenin also, there were alleles which have very positive effects, low molecular weight glutenin also, but not gliadins. The gliadins, they did not have the positive effect. So these 2 categories of genes, glutenin subunits, they were known to have the high effect on the quality, but the trypsin for the first time we showed here that they are as important as the glutenin, because they also form the polymer, they participate in the polymer formation, and as a result they contribute to the elasticity. Only thing is that these are not good for nutritional quality. They are rich in proline and glutamine, and also some of them are associated with the celiac disease also. But this trypsin is just like any protein of legume or any rice protein. And this got very high nutritional quality as well as it has the effect on the positive effect on the bread making quality. You can see here. This is the original HD2329, and this is one of the nils for the trypsin. The lobe volume increases here. This is a significant increase actually. And then HMW also increases. So its effect is as big as the HMW, because for several years the high molecular weight glutenins, the subunits 5 plus 10, they are very much glorified that they have very high effect on the quality. But this is globulin type protein which was first identified, in 1985. It has taken almost 30 years to know that this has also got as much effect as that. Unfortunately, not many people are working on this protein, but this is also published. Now, this is my last slide. Then what is the prospect, what we can do with the trypsin? We have done a lot of work on the variation, varietal variations and all those things. So we identified some alleles. So at least what we can do, we can take these alleles, superior alleles, and transfer in our best varieties so that you can have a better quality of wheat. Mostly the Indian wheat varieties are not very good quality because they don't breed for quality. You will only get what you breed for which you select. If you don't pay attention, that's why our high yielding varieties of Green Revolution, they don't have the climate resilience because we never selected for that. They don't have quality because we never selected for quality. We only selected for yield under high input condition. So they produce yield under high input condition. But under the stress condition, they don't even produce high yield. And sometimes we have selected for disease resistance also. So those things, you will have only what you select for. So nature selects under natural condition, natural selection. Breeder selects under captivity. But whatever trait you will put the selection pressure select, that's what you will get. So this is the time that to pay attention on the quality aspects of these things. So more and more genes we discover for the quality, and they could be put into the high yielding varieties, whether it is the processing quality or it is the nutritional quality. So, but there could be some limitation here because the allelic variation may not be that much, because the prolamines are the predominant protein. So that's where the role of genetic engineering comes. Like through genetic engineering, we can shut down some of the gliadin genes and give more copy of the trypsin. So one can have actually better lysine content or tryptophan content. So that may not be possible without resorting to the genetic engineering or genome editing kind of tools. So with this, I will like to close here. And thank you all for your attention. If you have any question, quick question, I will be happy to answer.
So now the talk is open for discussion. If you have any questions, please ask. Please come.
Thank you, sir, for this informative talk. I want to ask that what could be the reason of appearance of bands for this wheat protein on the gel where you are excluding mercaptoethanol, and what motivated you to come up with this idea?
Yeah, I told you there that these bands, they appeared because we excluded the mercaptoethanol. Normally when you do the SDS page, you reduce the sample. You will be adding SDS as a detergent to break the hydrophobic interaction. Sometimes you add urea also to break the hydrogen bonds. And then you include the mercaptoethanol to break the disulfide bonds so that you will get individual polypeptides. But I excluded mercaptoethanol because I wanted to analyze the monomeric protein, which is gliadins, because they are not linked with disulfide bonds. So you saw that when I excluded, I was able to see the gliadins without interference from the low molecular weight gluten, which is the similar size. But then we noticed these bands, and they were visible because they are linked with disulfide bonds. Their monomeric form, the original polypeptides, they also overlap with the gliadin and low molecular weight gluten. So that's why earlier we were unable to see it. But by excluding the mercaptoethanol, we could see that tetrameric form of these proteins, which was separate from the gliadin and glutenin. And because of the streaking which you get is very uniform, it's like a cloud. And on that top of that streak, you could see these bands. That's why we saw them first time. So I never planned for this, that I will see them. But I was excluding mercaptoethanol simply because I should be able to analyze the gliadins without having to do this cumbersome starch gel electrophoresis.
Thank you.
Anybody else? No? Sir, please come. So it was a mesmerizing talk. And by seeing your face, I can easily make out that you thoroughly enjoyed this talk. So sir has nicely presented all the dimensions of genomics and some of the success story like sequencing of mango or rice or peasant pea or tomato. So these are legacy we should cherish upon. Sir also explained some of the quality aspects and discovery of tryptophan protein. And there was, so this whole presentation was loaded with lot of quality-based informations and conclusion and suggestions, which definitely will give us idea to venture upon. So with this brief note, now I request Dr. Sally Praveen ma'am, Course Director and Head Division of Biochemistry, to please felicitate today's speaker.
So really, we are honored that you
Thank you
accepted
so
our
much.
invitation and you are here.
Thank you. Thank you so much.
Just few words I want to say. I am not going to sum up the talk because it is so fantastic and loaded with so much of scientific information. But what is the take-home message which I want to convey, which I gathered after listening 1 hour? The take-home message is, when we talk about our specialization, we say we are a biochemist. So we know more about proteins, we know more about enzymes. Then some of us, we say that we are biotechnologists. So we know more about genome, how to do sequencing, how to go for functional genomics. And there is a third category, we say we are the breeders. So we can only know about how to develop NILs or IRLs. So from today's talk, you can see 3 in 1. A person who knows much about biochemistry, who can talk about in detail about proteins, a person who is known for genomics, you can see a lot of papers in Nature and Science, right from 2000 onwards, so 20 years of journey in genomics, and a person who can talk very easily about breeding. So it is not about specialization, and it is not about that we are only in this area, we did PhD in biochemistry, so we cannot talk about breeding. So this is about your passion, how you can carry out your work in different fields. So this is the take-home message which I am taking from this class. And the second message is a person who started his career in first paper which we saw from the slide 1985, from starch gel electrophoresis. Many of us do not know because we started our life with polyacrylamide gel electrophoresis. A person who is talking so easily about starch gel electrophoresis to nanopore sequencing. So it is just the upgradation, and he is the man who is changing with time, not sticking just to starch that in our time it is we are talking only on starch gel. He can easily talk on nanopore sequencing. So that is the second take-home message, that it is not that there is, every time you can learn, and whatever new things are coming, you are adapting to it. So that is the beauty of today's talk, that I gathered these two messages, starting from one angle to another angle. And throughout his talk, not only when we say information, then second is knowledge. And throughout his talk, he talked about wisdom. Throughout, he is also giving message, when we are working, we are gathering information to convert to knowledge, and if we are not translating for the purpose of mankind, that is what wisdom is. So sir, it is a beautiful combination of science and wisdom. That why you do certain experiment, why you will spend so much of time and energy to do particular type of experiment, unless and until it is coming up with some purpose for some betterment in the mankind. So these three messages, what I gathered, I wish that at the end I will share with you that you also agree with me that such a illustrious talk from starch gel to nanopore, I have never listened. Thank you so much, sir, for coming and giving us this opportunity to listen to your whole 1985 till 2020 work in one go, in 1.5 hours. Wonderful. Thank you so much.